Welcome to our Expert Insights here at Industry Stage in Hall 11 D72 at Formnext 2023. I'm very happy that you're there at the live stream and here with us at the booth. And I'm very happy to have the hottest topic of the exhibition right now here. We are talking about emerging technologies, and where we cover topics like bioprocessing, like food printing, pharmaceutical applications. Our next speaker has a clear idea of the future, a future where biofabrication of organs and human tissue is common, and it's common medical practice so humans can enjoy a longer, healthier life. Dr. Tobias Lahm, join me on stage.
Hello.
Tobias, good to have you here. Dr. Tobias Lahm, he's head of technology of the startup Cellbricks, and he has a presentation brought with him, the future of organic biofabrication, multi-material, light-based bioprinting. Tobias, we are very curious about what you brought with you, and the stage is yours.
Thank you very much for this kind introduction. So 3D printed pharmaceuticals, that's already the future, I guess. But in some cases, medicals like tablets or pills won't help you and your organs fail. And what can you get then? I mean, there's treatment out there. You can get maybe an organ from a donor patient, but then this again is connected to lots of difficulties. So why don't we use this technology we have in hand and bioprint organs? So what you see here is basically a bioprinted tissue. And it is, in this case, a cube shape with an intrinsic channel system, so a vasculature. And what is different to usual 3D printing that you see here on this stage everywhere? Well, it's biologic, meaning there's cells inside and it's a living tissue, not only with one single material, but a complex structure based on many different cell types and many different materials. Luckily, we are on an additive manufacturing exhibition here, so I don't need to explain how 3D printing works. But in our case, we use stereolithographical bioprinting, meaning that we expose light to a stage and then a layer is solidified. The material is not metals or plastics, but it's bioresins, and these usually are composed of proteins so that cells can live inside. And these called bioinks don't only consist of proteins, but they have cells in there and also other additives that the cells need to survive and basically differentiate in their regarding tissue. So now we can combine different materials, different cells into complex shapes. And this is how we create basically living tissue since 2016. Cellbricks was founded in 2016 as a spin-off from the Technical University Berlin. And since then, we have done lots of research on tissues through all over the body, talking about vascularization, which is more or less the holy grail of tissue engineering, but it's a crucial step to basically get full-size printed organs. Also soft tissue for breast reconstruction, or functional tissues like the complex liver, which is a special case I will talk about later on. We are a strong team of 12 people and basically hiring more people as we advance on the journey. And we have a vast know-how in bioprinting and lots of partners with industry and academia to basically tackle this huge challenge. The technology has been proven in zero-gravity flights, which is for stereolithographic printing quite hard because usually it's an open system. So, and if you lose gravity, everything will flow around and you lose basically your cells and your ink. In this case, we enclosed the system fully, basically made a complete sterile closed system to tackle this challenge. And this is how our rack looked like, the bioprinting platform on there. And then we were flying in zero gravity and in 2G phases, and the technology worked fine. So we were able to print organs, which means in the future, if we go to Mars and everything, we can also basically have printed organs and tissues for therapeutic application. But how do we get there? I mean, right now you've seen some small tissues and they can be used in labs and they look like this. These are small fractions of tissues, basically the smallest functional units of a living organ. If you talk about the liver, then this is a liver lobule. If you talk about a blood vessel, then it's a piece of skin with a blood vessel inside. If you talk about the heart, then it's maybe just a muscle that you can analyze in lab. But these tissues are already vascularized, meaning if we scale them up, we can ensure that nutrients and oxygen gets into the tissue and the cells can survive inside the tissue. Because when you scale up something, the middle of your large object will usually just die off if you don't get nutrients and oxygen in there. To achieve these vascular channels, we created a software that is using parametric design to basically, according to biological function and to biological needs, create vasculature for you. You can do this by hand, but it's a quite tedious work. So having a software that is basically generating these vascular structures for you is coming in very handy. And then these channels and vascular structures can be printed with cells, and they are perfusible right after printing. So what you see on the left is basically one of those tissue samples, and a surgeon is putting blood in there. And on the right, you see that the complete channel is basically filling up with blood. Why do we need this? Unfortunately, there's lots of diseases out there that let our organs fail, and not for every organ there is a replacement. If you think about the kidney, there's dialysis, which works quite good, and you can prolong basically your life for quite a long time. But if you think about other tissues, for example, breast tissue, and you have cancer, and some breast tissue needs to be removed, you need a way to replace this, basically. Usually, this is done with silicones or with very complicated surgical procedures. But if you are able to recreate this tissue patient-specific, then this would be a really great win. And this is exactly what we are doing. So we basically have an application for post-mastectomy breast reconstruction to bioprint breast tissue patient-specific so that you can reconstruct the breast after surgery. This is based on a study that was done with one of our partners, and basically breast tissue was printed in this case. So we started with small fractions, having adipose-derived stem cells in there, and blood vessel cells, endothelial cells. And these will differentiate in the construct to fully differentiate to fatty tissue. So what you see on the middle bottom image is basically lipids and vesicles that are forming. And this is a sign for fatty tissue formation. So what is happening right now? We scale up this production and basically come from these small samples that can be used in lab to full-size breast tissue to help basically patients. And the next year, we will start an animal study with this tissue to make sure that all regulations and everything is fit. And from there, we go to the clinics and basically make this a therapeutic product. Let's talk about liver. The liver is very complex, but it's also one of your most important organs. If the liver fails, you basically die. There's about 1.5 billion people with chronic liver disease. These are liver diseases that are basically there right after exposure to different drugs, to different to your surroundings, and they are gradually. So basically, the liver doesn't fail right away, but it starts to fail and gets worse and worse. There's also liver diseases that you have right from the beginning of your life, so when you're born. One of 10,000 births per year have rare liver diseases with a genetic metabolic liver disease and a genetic defect, meaning the liver is there and functional, but, for example, one specific enzyme is missing and can't fulfill its function and can't detoxify your blood. So basically, you get a prototypical disease trajectory. The liver loses function over time until you reach the threshold where you actually need a donor organ to survive, or the patient would die. There's no universal platform right now to do any therapy on there, and the time to the liver transplant is very critical. Even if you get a donor organ, then this is connected to huge complications. For example, you have to take immune suppression because the donor organ isn't actually yours. So you have another organ in your body. So luckily, Cellbricks has done lots of research on liver tissue. So these are bioprinted tissues with different cell types. We started with cell lines, what you usually do to basically establish a new technology and a new procedure. But now we also use primary cells and induce pluripotent stem cells, meaning these cells can be differentiated and there's universal donors out there. So you can take these cells and these cells fit to most of the people worldwide. So they are readily available. If you take these cells and cultivate them in a 3D print, then you basically get a tissue that is functioning like the liver. We showed this in a couple of studies in vitro, meaning in the laboratory, and this is what you see on the right side. This is data showing that this tissue is expressing proteins and enzyme-specific, liver-specific enzymes over a long time. If you compare this to usual cell culture, which is, for example, used for pharmaceutical research, then these cultures are not only viable for a longer time, but they are also more active and more accurate for a longer time. So if you think about the prototypical disease trajectory and we diagnose a liver failure, which is chronic and which is based, for example, on a specific enzyme loss, we can take these universal donor cell lines, we can bioprint a tissue patch and basically implant this tissue patch into the patient. The tissue patch will basically recover the liver and take over the function that is missing from the native organ. And then we can heal the patient before there's even any donor organ necessary. So what does the future of biofabrication actually look like and what does it need? You've seen some examples now, and for example, for the liver, we also go into in vivo studies next year, meaning there's animal studies to ensure efficacy and safety. And after that, we go to the therapeutic application, meaning a scale up and clinical studies. What we need, therefore, is what we built up over the last years. It's basically the platform we use to create therapeutic tissues that are bioprinted. It's first of all a tissue engineering software to create these complex shapes. Partly you can use CT scans or MRT scans, but you have to basically make them better and make sure that all the blood vessels you need are there, or blood vessel sprouts. Secondly, you need cells that are preferably readily available. I mean, you can always use primary cells, meaning from the patient that is used, you can take some cells and you can transplant them from one region to another. This is, for example, done right now for breast reconstruction, but it's very complicated and it's not an endless cell source. So if you have to replace a large tissue, you just can't get the cells. But induced pluripotent stem cells have the capacity to be expanded, and you can use these cells to basically tailor to a specific patient. And then the bioprinted tissue is more or less not a foreign tissue, but it is the patient's own native tissue. You also need clinical biomaterials. So this, of course, is not plastics, but it's the protein-based bioinks I talked about. And these need to be not only powerful on a technological way, so they don't need to be performing in the bioprinting process, but they also need to be basically good for the cells, let's say. The cells have to grow inside of these hydrogels, and they also need to differentiate in these hydrogels. And what's most important, they shouldn't get cancer. So if cells are in a foreign environment and they start to proliferate, like they double each time, they tend to get just cancerous, meaning you have tissue, but you have bad tissue. That's not good. So if you put together the cells and the biomaterials and you have your 3D model, then we can bioprint this in a GMP-compliant bioprinter. As we use light-based bioprinting, which is more or less stereolithography, we can have very high resolution and we can have very large objects. So we use a DLP-based process, meaning that each layer is projected at once. So like each plane is growing, let's say, at the same time. And then afterwards, we have strong connections to clinics, of course, to basically get these tissues into patients. Because after printing, there's a maturation phase in the laboratory, but at some point, it needs to go into the patient to be viable and to survive for a longer time. So basically, we as a human body are the best incubators you can have. No matter how high-tech your lab is, the body is always best in keeping cells alive and keeping tissue function. This is my team, basically managing right now our growing phase, and I'm very thankful that they are there and leading us there. Alexander Leutner, who's our Chief Executive Officer, Joachim von Arnim, who's doing the strategies, and Dr. Lutz Kloke, who has founded the company back in 2016. And we have our head of implant, which is Anna. She's basically leading right now the breast tissue reconstruction and the program therefore. And we have Alexander Thomas, who is our scientific lead and managing the clinical studies for us. And of course, all the rest of the team. Without them, it wouldn't be possible to get the work done. Thank you so much, and let's talk afterwards.
Yeah, thank you very much, Tobias. Thank you very much for the insights. I mentioned earlier our Slido tool. So we have a first question, but before I want to show you where it is. There is a roll-up over there. This slightly dark roll-up over there. There is a QR code, and here you can join the conversation, or here you can post your questions here from the booth, and at our live stream. There you will find a similar QR code next to the live stream window. I hope it works. Yeah, and Tobias, as I said, we will have the first question for you. What material was used to print the liver tissue engineering scaffolds?
Yes. So what is established in bioprinting is to use gelatin at first, because it's readily available and it's performing quite well. So the first constructs we did were based on gelatin, which is then methacrylated to basically make it photoreactive. Nowadays, if you think about primary tissue, and especially going into therapeutics, then you would try to use xeno-free material, meaning gelatin coming from a pork, for example. You don't want to have this in your body. You don't want to have animal-derived substances. So we go to recombinant material, which is fully defined and basically synthesized by using microbiology and biotechnology.
So how do you define which materials you use? Is there, are you always talking to doctors, or how does it work in your team? Where is the most recognized specialist there?
Yeah, well, I mean, this is all based on biomaterial engineers, but it usually comes from the cells. So the cells tell us what material they like most. They need to attach to the material, and they need to have specific function in the material. And then, of course, if you think about the application, this basically defines whether we can go with a material that is based on animals, or if we go with a material that is engineered for medical application.
Mm-hmm. Okay, how long did the cell tissue survive, and have you tried a bioreactor to increase this time?
Yes. So the first studies were based on a 14-day trial. This is usually what you start with. The cells are viable and they are prolonged in life. Our longest bioprinted constructs have been in culture for, I guess, 60 days. And then we basically stopped it because they would renew themselves all the time, so potentially they would live forever. Using bioreactors is a very good idea, especially if you think about application in the lab. So if you want to have a tissue that you can do pharmaceutical studies on, and you want these studies to be longer than, let's say, a couple of days, then perfusion comes into mind. So you could use organ-on-a-chip systems that then perfuse these channels and also trigger the cells and basically, let the tissue survive for a longer time.
Mm-hmm. Okay, one question from myself. When you said that you are working on the lab, so I think the printers you're using there, are they already so that you said, okay, we can transform them into some industrial area, or do you need maybe some more work on it to really make the printers itself industrial scale?
Yes. So right now we have a couple of generations of printers. We have all built them in-house or with partners together, but they are basically ready for these therapeutic applications. So what they are not ready maybe is for a mass market, because it's a very specific field and a very complex procedure that is highly regulated. So each printer is very specific according to the lab that it is used in. Okay.
Now what I'm looking at, some additional questions. For the breast reconstruction, how will breast implant be attached to the patient's arteries for vascularization?
Yes. Very interesting. So usually, if you do breast reconstruction, you would use fatty tissue from other parts of the body. The good thing about breast tissue is that it's not highly vascularized at first, or basically the vasculature will grow in there by itself. So you don't have to do anastomosis, basically like sewing each blood vessel to each other, but you need a tissue that is quite perfusible. So let's talk about it like a sponge. And if you have this, the blood will basically flow in there and blood vessels will form by themselves.
And that's the same method you use when you create other organs. You mentioned that there have to be oxygen inside, that there have to be nutrients inside. So that's the same principle you always
Yes, definitely. What we do is we print with a resolution around 30 micrometers. So this is basically the vessel sizes we can create, or we can get starting points. But microvessels can be a lot smaller. But then we let biological do the work. So basically the endothelial cells, they will form these vessels if they are needed. So if there's a larger construct and there's no vessels yet, then they will sprout in there because there's a need for vessels to be there.
Okay. And you mentioned another question from me, not from the audience, would be, you mentioned going to Mars would be a good point because you can create some new organs there. Why did you to be serious? Why did you do these experiments in low zero?
Yeah, there actually is a need for spaceflight for therapeutic applications. The most injuries that occur in spaceflight are burning wounds. And these burning wounds can be quite large. And what is done today is you put patches on there to basically cover these wounds, and then you expect the skin to reform themselves and to basically have blood flow in there, and so that the healing procedure will start. But if we print such a patch with specific cells for the patient, you can basically have the exact size, you can put it on there, and it's not healed instantly, but you have basically the best foundation for a native skin you can have.
And which material would that be?
This is again based on collagens and laminines, so basically all the materials, let's say, that are already in your skin, and they are basically done one-to-one to your skin composition. So it's mainly collagens.
Mm-hmm. And do you add, when you're talking about the tissues, do you add medicine as well? So can you apply that whilst you are applying your
Yes,
your
definitely.
new skin?
The cool thing about bioprinted hydrogels is you can control the pore sizes, meaning if you have substances in there, you can also control a release. So if you have to add some medicine in there, then you can also release over how many days, or control over how many days this medicine is released, and then like locally on the skin or in your tissue.
So that's very much personalized as well.
Definitely, yes.
Yeah, thank you very much to have you here on stage and for your time being with us. I have now some announcements to do. We will see you later here on stage when we have our panel discussion, and then we can dive deeper into the topics. And maybe you have the one or the other question for you as well. So we see you in some minutes.
Perfect. Thank you very much.
Thank you very much. Yeah, dear audience here, yeah, applause for sure. Thank you. Dear audience here at the booth and dear audience out there at the live stream, let me give you one hint here. At the area at the industry stage, we not only cover the topics here on stage, but we additionally have a cafe, industry stage cafe, just opposite here my stage, and there you can find some job announcement. There you can find new contacts if you are looking for a job in additive manufacturing. I think that it's very good starting point if you come to the exhibition, and if you want to go around, just start here in Hall 11 at D72. Yeah, dear ladies and gentlemen, here in the booth. What else do we have? Upcoming topic today will be, next here on stage will be our expert panel. I will give you a short outlook who will be there. Katie Zinze, she will be there. She's from 3D Additive Media. Terry Wohlers will be with us. Very interesting. He's head of advisory services and market intelligence. He's coming from STAM. They had a conference yesterday already, so maybe he will be. He has not a jet lag here when he comes to us. And some of you already saw Dr. Anna Worsley. She has been here just before, and Tobias Lahm from Cellbricks, he will be on stage as well. So make sure you don't miss our panel discussion. And very interesting, this evening we will have some startups. Tobias was from a startup as well, so we will have some startups this evening here on stage, and there will be some new prizes for the startups. And you know, they have a very short presentation, something like an elevator pitch. It's very interesting for you to have a look at these as well. Maybe you find it interesting. Yeah, that would be our day today, but we will go on tomorrow and until Friday. Just let me give you another hint about our program tomorrow. Tomorrow we will have the partner countries here on stage. Maybe you know, or you heard already in our morning talk, that we have the partner country, not just one. We have a real area. It's the Scandinavian countries. We have Finland, Sweden and Denmark coming to stage tomorrow. And right next to our booth here, we have the special area, the pavilion, although there is no roof of the pavilion, but there is a pavilion of the partner countries from Scandinavia. Yeah, dear ladies and gentlemen, I'm very happy that you're here, and I'm very happy that you are following us. Now I would say we have a short break here, a cup of coffee, and we will meet on time. Again, let me have a look at 11:40.